No, I think I wrote too much stuff, so she was like, oh, I can let her finish.
Okay.
Because I wrote like things.
What was that? Oh.
I'll go through it and
I just made a few sentences. I learned it like a minute.
Okay. So for those just showing up, you're lucky. Okay. A few things. Lecture 8. Remember we were recording it and a thing came up saying I was running out of space on my hard disk and I was like, oh God, I hope it's okay. It wasn't okay. I went to go convert that, to export that file and it can't be read. So I'm sorry, but you can use last year's lectures. It should be around lectures 13, 14, or 15, so luckily. Huh?
Yeah, it's on Blackboard right now.
Yeah. So sorry about that. The other thing is I have, for those who haven't got their exams yet, I have them up here. So just come up after, you can grab it. The other thing is, someone asked me today if the next exam is on time or not. I'm going to actually go through and figure out if it's going to be on time or if I'm going to have to change things up again. Sorry. I will let you know ASAP. Okay. So far what we've learned, we just introduced cell cycle, talking about the phases and the checkpoints. We talked about different methods for study, BRDU, pyridium iodide staining, temperature-sensitive mutants, just looking at the cells, staining with antibodies, immunofluorescence. We spoke about how CDKs are regulated because CDKs are the kinases that phosphorylate everything involved with cell cycle regulation. So we spoke about cyclins, WEE1, CAK, and CKI. We spoke about the ubiquitin proteasome system, and specifically we spoke about APC and SCF, ubiquitin ligases, which are your two major ubiquitin ligases involved in cell cycle regulation. Oh, that's not a good spelling. And then we talked about S-phase. I don't know if it's that I teach from 10 a.m. to 5 p.m. on Thursdays, or if I'm getting sick, but my voice is going. We spoke about S-phase in specifics, right? We talked about regulation, how S-phase is kind of set up with the pre-replication complex, or pre-replicative complex. We spoke about how actual origin firing is initiated by SCDK. We spoke about steps in DNA replication. I introduced the trombone model, and in recitation, you guys have thought about it for a long period of time. Hopefully, it's all making sense. And then we spoke about the mechanism of DNA polymerase, how it actually works. There's a lot of question in recitation about 3' and 5' and leading and lagging and parental and this and primer and that and whatever and what's going on. And I think those are a lot of places that are really easy to get confused. But I find that if you remember the simple rule, which is that DNA polymerase cannot make DNA out of nothing. It can only extend DNA. And it can only extend DNA by adding to a 3' OH. And if you remember that, you'll always know in which direction things should be going because you're adding onto the 3' end. So you're always going 5' to 3'. Okay? So if you can remember that and understand that, then you should be okay with the directionality of things. Okay. Does that make everyone happy? Yeah? Okay. Okay, so we ended up talking about fidelity the other day. And I was telling you how fidelity of DNA replication, so fidelity meaning staying true to the sequence that is in the parental strands of DNA. Fidelity is maintained at 2 different stages. One, during polymerization, because the correct base fits in the enzyme in the active site better than the incorrect base. And I'll show you a few examples of what I mean. Because remember, in that active site, you also have the base of the parental strand. So there's a certain amount of space taken up by that base. Not the base, but like the strand and that base. And then you also have the incoming base. And it's not going to fit as well if it's incorrect. So that's one thing. But still, there are errors. And so after polymerization, you can get removal of an incorrectly added base based through the proofreading capability of DNA polymerase. Okay, so what am I saying? It doesn't fit well? Well, so if you think about how it should look, so the white outline is how it should look, and an incorrect base pairing is the shape that's overlaid in the pink. And you can see how, in terms of the amount of space that's present in the active site, there's ideal shapes. And if it doesn't fit properly, it's less efficiently added, ideally. Okay, and so this is just showing like it should be GC, but if you add a T instead, it's going to kind of look like that, and so on. So that's one thing. But most importantly is this 3' to 5' exonuclease activity. And so, well, yeah. And so when I talk about fidelity and I talk about this 1 in 100,000 error rate, it's mostly due to this exonuclease activity. Not to say that errors don't happen, and we have repair systems for that, but this is one of the reasons that the replicative DNA polymerase has such high fidelity. So, so if you look at your hand again, and in your palm is your active site, down here is your exonuclease active site. And so you're going to start thinking about domains and how there are different domains that make up proteins. So this could be is probably an appropriate name, but this would be like your DNA polymerase domain, your polymerizing domain, or something like that. And this would be your exonuclease domain. So it's protein with different spots that do different things. So, so in the off chance that a incorrect nucleotide is being added, and it's being shown here by that little pink dot, which hopefully you can see easier at home than on this. That little pink dot being added to the yellow or greenish newly growing strand, that's being added incorrectly. And so what happens is it's moved, it's kind of stalls, and it's transferred to the exonuclease active site, and it's removed. So it actually kind of backs up, yeah, and removes it, because it's 3' to 5' exonuclease activity. And then the end is transferred back to the polymerase site, and hopefully the next base that comes in is the correct one. Okay, yes?
So that's the polymerase, right? That's the polymerase and the exonuclease inside the polymerase?
Yes.
Oh.
Yes, yes. So it's an exonuclease activity within the DNA polymerase. Okay? So it has its own domain.
Yellow is the new strand, right?
Yellow is the newly growing strand in this picture. Yes. Yes.
I'm lost with the citation. So would the yellow strand be the looping thing?
Don't worry about this, because this is not showing leading or lagging or anything. This would happen anywhere DNA polymerase is working. So you have a polymerase on your leading strand, you have a polymerase on your lagging strand, right? No matter what, because you need that to add on a basis. And just all replicative DNA polymerases have this exonuclease ability, enzyme activity. Yeah. So the question was about like in the looping out, how does that all come together? Remember, with that whole thing that you did in recitation, DNA polymerase was there, and DNA polymerase was doing its thing, and this activity is within that DNA polymerase. Okay? Any other questions? Okay. so this is just going through it more slowly, right? So the rate of incorporation of the next base after incorrect incorporation is slow, and so that allows for that shifting of the strands into the exonuclease activity site and removal. So it kind of slows things down. So this is proofreading. When you talk about proofreading, this is it. And so it's actually part of DNA polymerase. It's not something else going on. Now, yes, there are proteins in the genome that are checking for the correct structure of DNA and recognize errors or DNA damage in general. But when we talk about proofreading with DNA replication, we're talking about exonuclease activity. And so that basically means that the incorrect base is recognized because it's not base pairing properly, it's the wrong shape. And then the strand that has the incorrect base added is transferred to the exonuclease site where it's excised, where it's removed. And then it's transferred back to the active site for addition of the next base. Okay, sometimes I'll say NTP, that's just nucleoside triphosphate. So you know there's like all the different bases, right? ATP, GTP, CTP, right? So NTP is just the general term. So if it shows up on an exam or on a quiz or on something, just kind of keep in your mind that means base, okay? NTP equals base. Okay, so now let's think about HIV for a second, because people love to talk about HIV. Well, I do, at least. So when you think about HIV and you think about drug resistance and how drug resistance develops so quickly, and it's so hard to create a vaccine, well, that's for multiple reasons. And you think about just how, in general, this virus mutates at an alarming rate, which is why when you're treating someone with HIV, usually you give them a cocktail of multiple drugs rather than just one, because then you're hitting the virus at different angles to keep replication low, so resistant mutants can't arise. But anyways, the reason the mutation rate is so high is because reverse transcriptase is error prone and has no exonuclease activity. So it's just a sloppy way to replicate the genome. So the error rate with HIV's RT reverse transcriptase is 1 in 2,000 to 1 to 4,000. Compare that to DNA polymerase, which is 1 in 100,000. So every 1 in 2,000, let's say 3,000 in the middle, bases is incorrect when HIV is replicating its genome. Incorrect. So that's like huge chances for mutations to arise.
So the mutation will be incorporated.
Right, so the mutation, there's nothing to fix the mutation, so it's incorporated into the new virus. And so then you think about selective pressures of a drug coming in, right? But aside from anything, if it's a mutation that's going to make the virus not survive, then who cares? But if the virus is fine, and then that virus is resistant to a protease inhibitor or some kind of drug, boom, you have these resistant viruses. Okay, so we should be very happy we have our exonuclease activity.
Is it possible to like inject a functional gene for DNA polymerase that has that exonuclease activity to HIV?
Is it possible to basically provide exonuclease activity to HIV? So why would you want to provide exonuclease activity to HIV?
So you could just use one kind of DNA instead of just like combining multiple drugs. Just use one and just.
So you're saying to cut down on the error rate and decrease the possibility of mutation, why not?
Focus on one drug that will-
So that you can have one powerful drug and you'll never have resistance arise. That would be fantastic, except for the fact that like out in the world, this virus would never survive, right? So it's like, you know what I'm saying? So if you want to put something out into the environment, it needs to be something that has a high enough fitness to kind of survive. Whereas HIV, now I'm sure someone's going to listen to my thing here and say, but HIV, like there's a huge selective advantage, right, for having a high mutation rate when you're a virus or a bacteria or anything, right? Because if you can keep dodging the bullet, you're going to survive. Whereas if you give them a nice exonuclease activity, then they're going to be like, well, we're dead. So, yeah. Yes.
Wouldn't that make a good target though? If you targeted cells that didn't have the exonuclease?
If you target,
Because like doesn't their
When you
viral
provided the
polymerase-
exonuclease activity and then you have one drug.
wait, let's see what he's saying, and then you can jump in.
Is
Yeah.
it like the viral polymerase doesn't have an exonuclease?
So the so the polymerase that HIV has is reverse transcriptase. It's encoded by the HIV virus. Yeah. Yeah. What were you gonna say?
No, I was just trying to continue with what Charles was saying about make sure that they will not gain the resistance, so providing the exonuclease activity and then have just one drug.
Well, yeah. So that would be great in the lab, but not in nature. Right. Great. Okay. Any other thoughts, comments? Anyone else want to talk about HIV?
Are they using two different templates? So is HIV using like the RNA template and we're using the DNA?
HIV has an RNA genome. Yeah, so that's why it has reverse transcriptase. Yeah. Okay. Anything else? Take Barry Margolese in virology class. Learn more about HIV. Okay. So now let's move on to enzymes, these enzymes that are involved in DNA replication. So you guys know them all already because this week in recitation we drew out our replication fork. Today we drew out our whole replication bubble, which was quite an exercise. You saw all the enzymes that are required generally. We didn't include them all because we were kind of just focusing on the fork. But what you already know from your drawings is you've got your polymerases, you've got your helicase, you've got your primase. Where are they having? They have primase right there, bound to helicase. Ligase, which I don't think they have here because that ligase is what's going to kind of sew up all those Okazaki fragments, and topoisomerase. So we'll go through all of these enzymes. Okay, so first let's just talk about polymerase. So I was saying to people today in class, and just I think I mentioned it before, there are different types of polymerases. It's not just DNA polymerase. You can think of them as two classes of polymerases. Processive polymerases, and so they can go for a long time without falling off, and distributive polymerases that fall off easily. And they're going to serve different roles, right? If you're copying your genome, you want a processive polymerase because you want to go long distances without falling off, especially in your leading strand. And if you're just repairing your genome, so little bits of DNA that have to be repaired, you're going to have these non-processive or distributive polymerases. So this is kind of just showing the two of them. With processive synthesis, you're just going to keep going. You're not going to fall off. With distributive synthesis, you're going to kind of synthesize a little bit of DNA, fall off,
So isn't it what you need for the
bind again, fall off. Is it what?
No, I think I
And so it's this constant bind, fall off, fall off. And that's useful if you are doing like base excision repair, nucleotide excision repair, some of these repair mechanisms where you need to cut out a chunk of DNA and then you use the template strand to repair whatever damage was there. And we'll talk about it a little bit more during the end of the semester. So there are different types. Yep.
Even though DNA polymerase, like there's
Yeah, because, okay, so you're thinking in terms of the lagging strands, how you're kind of binding and then coming off. Still, it's relatively long that it needs to copy. And, that would be considered recessive, because it's the same polymerase that's doing your leading strand. Any other comments? Yes.
Structure-wise, leading DNA polymerase, it's same as the lagging DNA.
Yes.
Like, there's
It's the
no-
same DNA polymerase on your leading strand and your lagging strand. Yep. Where these other ones come in are usually DNA repair. So here's the different types of polymerases. You've got your replicative polymerases, so these are the ones that copy your genome during S phase, and that's what we drew today, that's what we're talking about mostly right now. Then your, and so B, C, and D is DNA poly, B, C, D. And then you have your DNA repair polymerases, bulky adduct bypass, translesion synthesis. Those are DNA polymerases that will come and bind when there's DNA damage and bypass the damage. They've got like this gigantic active site that can fit all sorts of damaged DNA, and that's only done when it cannot be repaired. Sometimes it's better to bypass the damage than to, because then the organism survives, right? When in doubt, translesion synthesis. RNA primer removal and gap repair. So again, remember, you've got your RNA primers. They need to be removed, repaired, and then ligated. So that's DNA polymerase A or alpha. And then there's reverse transcriptase, which copies RNA to DNA, and that actually is telomerase. And so those are your major types of polymerases that we have. There's a list. And so there are different families. It's pretty much what I just said to you. Okay, so let's move on to helicase. So today when I was watching everyone drawing, I noticed a lot of people like to draw the helicase around the double strand of DNA. And hopefully what you learn through the exercise is actually your helicase is wrapped around one strand of DNA, and that's how it unzips the double strand DNA. And so we'll go through that. But so this is just showing, it's actually the crystal structure of HPV's helicase. HPV encodes its own helicase. But still, it's representative of helicase. And so this is kind of showing how it would be sitting on the DNA. It would bind to one strand and move down. And if you look at your zipper, that's how your zipper works, right? In a way. Yes.
So is it always a life strand?
I believe everything I've ever seen, question is it always the lagging strand that it binds to? Everything I've ever seen has always had helicase on the lagging strand and moving down forwards. But I don't want to say 100%. Yes, but I've always seen that. Yes.
That part just confuses me. If the helicase is part of the whole enzyme, and helicase is the one that has to open up the DNA, how could it be one unit but still going ahead? Because it has to act first before the polymerase can actually act.
Let's see if we can get there. So let's go through the rest of the stuff about the helicase and see how it works with you. But it sounds like what you're getting to is how are they bound together and working together?
Kind of like the
How do
way
they get
we were
on?
taught before made more sense to me, that helicase goes first
Yeah.
and then the rest follows.
Yes.
But if all of it is just one enzyme.
No, well, it's not one enzyme. Okay, so I see where you're going. So the question is more about like how are these proteins organized, okay, and how are they working together? And so in what we drew today, here, let me take a minute. and then we'll come back to this. I love when people leave paper here for me. Okay. So in what we drew today, right, you had your, let's see if I can give myself enough space. That's your double-strand DNA. And so then you can have your helicase around here. And yes, it's going to be bound. Is that working? Yep. It's going, so there's an H for helicase. It's going to be going first because it has to open up the double-strand DNA. So it's going to be moving in this direction. And it's going to be interacting with DNA polymerase. I'll put a P. So that's your leading strand. And it's going to be interacting with your other DNA polymerase, which is your lagging strand. And so let's see, let's put in our primase, PR for primase, to make the primer. And then we'll have another primer here, right? And so this polymerase is going to bind to here and extend that. And it's moving that way. And so this is also, was already there. And so this is moving that way. So it's all moving together in one direction. And helicase is coming first in a way because it's opening it up first. Does that make sense? So is that what you were asking about? Because it is not one protein, it's one complex of proteins that are kind of acting together. So they're working as a unit.
So basically, every segment that the helicase opens is immediately basically polymerized.
Well, so on leading strand, so there's your leading. Yeah, because the minute it opens, polymerase is right there, and it can start kind of adding bases, right? And the lagging strand, it'll immediately add a primer. Well, and then it'll wait a little bit, and then it'll add a primer, then it'll wait a little bit, and then it kind of loops around, right?
Okay.
And so then once a primer, and so that's where I say keep in mind polymerase needs to add to a 3' OH. So once it hits the primer, remember your 3' OH is right here. And then it can extend off of it. Okay? Okay. Yep. Yep. As soon as I heard that, I said we need to stop because probably other people in this class could use that. Okay. Okay, so is everyone feeling, I know I don't have everyone for recitation, but is everyone feeling okay about how this all works? Yes?
Is the DNA ligase also on this complex?
No. DNA ligase is not on that complex because DNA ligase comes in later and joins things together. Now, let me take that back. There is always a possibility that it binds there transiently or something like that. But my understanding is that you've got your replication fork opening and you've got polymerization. And then DNA ligase, its job, and we'll talk about this in a minute, its job is once the primers are removed and the DNA polymerized in that section by the other DNA polymerase, alpha, I believe, then there's like going to be a little bit of a gap between the DNA that was made with the replicative DNA polymerase and DNA polymerase alpha, and then it comes in and kind of joins them. And so there's no real reason for it to be bound there and functioning there. Yes?
Should we concentrate on specifically which part DNA, like specifically alpha, binds to, like DNA alpha polymerase, is that how you say that?
I would focus on what your replicative DNA polymerases are doing and what your other DNA polymerases are doing. Because you can think of it as two. your process of DNA polymerases and your kind of distributed DNA polymerases. Yes. Yes.
So, a polymerase complex, like it would contain a polymerase, a helicase, a primase, and a
Not topoisomers.
Yeah.
Sliding clamp loader. I did not put those in. I was
Clamp loader is also part of complex or just?
yes. Clamp loader interacts with helicase. But I didn't put that in this drawing because I was just trying to address her question. Yeah.
but not ligase, right? You said it's-
I believe ligase is not permanently always there. It might have a binding site there where it might come and go or something like that. I'm not 100%. Because it would make sense that it would want to be close to. So let's just reason through this really quickly, okay? Because I might not know the answer, but I can reason through it. So if you're replicating your DNA, ligase activity would be needed where your Okazaki fragments are, right? So it's logical that ligase might bind to that complex, but it also, depending on how slow the activity of ligase is compared to the activity of polymerase, it may not always be bound, right? It might be in close proximity somehow. Does that make sense?
Yeah.
Okay. Any, yeah,
So on the illustration, where on that illustration, what is the design fragment? A large arrow that is on the loop or after that?
so here's the beginning of an Okazaki fragment. So let's say here's another primer. And so that polymerase is going to move in that direction, polymerizing until it hits that other primer, and then it's going to fall. Then it's going to release. And so this, from primer to primer, is an Okazaki fragment. Okay? Yes.
For how long exactly is DNA synthesis on the leading strand pause? I know it starts when primers are synthesized, but for how long it's being stopped?
So the question is, how long is leading strand paused while the lagging strand catches up? Is that what you're saying?
Like the whole loop has to be synthesized and then it starts
Yeah.
again? Or?
I'm not sure. That is my answer for you. Yes.
So is it safe to say that there's a primer before and after the DNA polymerase on the lagging strand?
On the lagging strand, is it safe to say there's a primer before and after? Yes, because polymerase is over here, so it would have had to start by adding on to the 3' OH here, and it's going to finish this one because this could be Okazaki fragment number 1 here, and this would be number 2 here, right? Okay. Oh God. I'm noticing a lot of things here. So yes.
So is it safe to say that conceptually the pause actually, like, based on time takes place between when your primase finishes putting on the primers and that pause then happens until your lagging strand binds to the rest of the polymerase?
So I think it has to do with the speed at which primase works. Primase is slower than DNA polymerase, and I believe that causes the pause. That was my understanding from that paper. Yes.
Okay, so that primer you have up there, it's gonna come down, and polymerase is gonna make the corresponding change. So when does the effect of the polymerase stop? Is it when it reaches a 3'
When it-
end?
When does it? So you're saying when does DNA replication stop?
Everyone is asking the same.
So this is great because really what happens, remember, so remember we did that paper where we're talking about timing and location of different origins in the genome, right? And so each of these is a replication bubble. This is happening inside each one. This is happening here and it's happening here. It's happening here and it's happening here, right? And so this replication fork's going to move this way. this replication fork is going to move this way. And so eventually what you're going to have is that. Right? And it's going to expand and it's going to expand and it's going to expand until it looks like that. Okay, so it'd be like that. Actually, I think I can. Looks like I don't need PowerPoint slides for this lecture. Oh no, but the people at home hoping for, none of this is recording. Sorry guys. Okay. This is a test. Yeah.
Oh, yeah.
No test. Okay, so is it all coming together for people here? Yeah.
Just have one more. So the trombone loop that we see is only going to be on the Leyden strings. And the loop is only created between the helicase and the DNA polymerase.
The
So
loop.
any material between those 2 points is going to be a part of the loop.
Any material between the helicase and DNA polymerase on the lagging strand is part of the loop. Sounds good to me. Now there is a secondary kind of primase loop that also occurs, but we're not going to go there. Any other questions? Yeah.
So the start of that period's primer and until the end of it, that's considered Okazaki fragment.
Yep. So this right here is an Okazaki fragment. From the start of a primer until you run into the next primer.
Single strand fragment, but we have to remember that.
Well, I mean, it's important to understand, and we're going to get to all this right now. It's important to understand that when you have single-strand DNA, it wants to fold back on itself, and so you need single-strand binding proteins, and that's actually in this lecture. We're going to return to. Okay, is this helpful for people?
Yes.
Okay. Are we okay with moving on? Okay. Okay, I know it's really hard because we're constantly zooming in, so it is important. Every now and then ask me, who asked me to, was it you? Every now and then ask me to zoom out so you can get that big picture because it's so easy to focus on a replication fork, which is quite detailed. Okay, so here's helicase. So now helicase is unzipping. This is only showing helicase, but if you were to add on DNA polymerase, they'd be associated, okay? Okay, the DNA helicase is a hexamer. Hex is how many? 6. Of one protein, so 6 subunits of one protein that forms a ring and uses ATP, so ATP-dependent ATP hydrolysis and associated conformational change to propel itself forward. So I was thinking to myself I should do something about ATP and ATP hydrolysis and how it affects movement. So I'm going to have to remember to put that in a lecture at some point because I feel like it wasn't until graduate school that I really started to appreciate what was happening there, or even when I started teaching, because you're told constantly, this is ATP dependent. Okay. What is ATP doing? Yeah.
And I've taught that because ATP has like the phosphate bonds which are really, really strong. When you break them, it releases a lot of energy.
Right, so I've been told that too, that it's got these phosphate bonds, and when you break them, that releases energy. That still doesn't mean very much to me. Does it mean much to you, or is it just something you learn? It's hard to visualize. I actually read somewhere, and this was more in relation to GTP, that if you think about the shape of ATP or GTP, we'll call it NTP, compared to ADP or GDP, what's bigger, ATP or ADP? ATP. Right. So you can imagine if you've got something, let's say I'm a protein and I'm like this, and I've got these like big donuts between my hands that are ATP sized. If you have ATP hydrolysis, and now I have ADP, I'm going to do that, right? It's going to cause a shift in my structure, that conformational change that we hear about all the time. And so, like, that might not be the most scientific way to explain it, but it makes so much sense. And so really, a lot of the time, you have, so like in this case, what we're doing is we're moving down, we've got our DNA our helicase binding, and it's going to propel itself downwards, and it's saying it's using ATP hydrolysis. And so that ATP hydrolysis is causing a change in the shape of the protein that's propelling, that's causing its movement. Now exactly how that occurs, I don't know. But you can imagine if you have 6 subunits, actually, does it say, I believe 1, 2, 3, I guess they must all be binding ATP. So if you've got 6 subunits and they're all binding ATP and they're all shifting, you can imagine how it would kind of create some kind of motion that might move it forward. I think it's important to try to think of these things. Yes? Is it constant hydrolysis? Yes. So the movement is, yes, so each bit of movement is another ATP, exactly. And I wouldn't even say one. I wouldn't be surprised if multiple ATPs, because there are all these different subunits coming together. And so that's what's happening. And so that's why it's important to understand it's around one strand of the DNA, and it's kind of coming between the 2 strands, and that's how it's unzipping. Questions? Okay, so we're going to stop and do a practice problem. I realized from the exam that I don't give you guys enough practice problems because on the short answer, people did great on numbers 4 and 5, which are things I kind of drilled into you, but couldn't apply it on number 3. And I think that's my fault for not giving you enough practice. So we're going to try to get more practice problems. Okay, so you are studying, so the question is protein X a helicase? Okay, so you're studying a protein with unknown function. You blasted it, because that's how we always start, and you have a sequence. And you found it contains a conserved helicase domain. So you hypothesized maybe it's a helicase. So design a biochemical assay or some kind of experiment to test for helicase activity. And so this is going to actually require for you to think about a few things, and I'll help you through it. So the things that you're given in the lab are some single-stranded DNA, and you'll get some long fragments and some short complementary fragments. When DNA is complementary, it binds, right? You'll get some ATP. You'll get a spectrophotometer with the knowledge that single-strand DNA and double-stranded DNA absorb light at 260 nanometers differently. And you'll get an agarose gel, so you can evaluate the size of your DNA. So I want you to take a moment to work with your friends and go and work on this. And I'm going to come around, and something I said to someone today was, sometimes when I do these things, it's very overwhelming for some people, and the instinct is, I'm just going to wait to get the answer. That's not going to help you get to where you need to be to do well on the exam. So as I'm circulating, call me over and be like, I've got no idea how to even approach this. Help me, and I will help you, okay? So, work together. This is a hard one, but an easy one all at the same time.
Right, put it like a desert or whatever. It's gonna bind, right? So it's gonna go far to it. Then you turn it to the helicase, and then you take that DNA from that helication. If it's to the left, then you want that.
So, the initial exercise, just take your double strand it's similar to like application But y'all,
And then this, I'm giving you essentially like,
why would you?
yeah, it's giving you essentially all of
Yeah, but it's gonna be the
You should see.
Yeah, but you have to start that double strand DNA in order for you to get things done. But that will give it after relocation.
Why would you do that? Why would you do that?
Yeah, but that's not really. So you start with a double strand DNA, right? You're throwing your single strand to complement the pieces of brain, right? And the second one, you can throw your complement DNA plus the suspect that it's helicase. But if it is helicase, then you can expect higher molecular
Unwind the DNA and that little piece of DNA would be gone. So it would be separate. You have like two separate lines.
Yeah, but those lines of mine will complement means that those people have more DNA double strand rather than having a single strand.
I don't, my knowledge is
No, give me an image.
I'll go.
This is what I'm saying. You start with double strand DNA. This is your there's no helicase. You're just throwing your complement DNA.
no, you start with a single strand. You're not starting with a double strand. You're starting with a single strand. It's not double strand. See, it's gridlock, a single strand. Right. So you have one big single strand and one tiny small complementary fragment. That's what you say. You basically have one single strand like this, and then you're throwing one little small complementary fragment. You don't get one single strand. If you look at the picture, it's not a full strand.
It's just like
You're starting to look whether it's a complement that's not, it still looks like a double strand, right?
That is the complement that you.
I know that, but it looks, if you ignore those side parts, but it is.
That would still be a single strand helicase when you get it. Because it's just helicase, it's not we don't think there's a risk complement.
Anyway, so if that complement, whatever that is, if it's not helicase, you can get double strand DNA.
No, you get the same.
Yeah, that complement.
Okay, guys. So there are some people who seem like they're on it. Some people who told me they don't even know where to start. So we're gonna go through this together shortly. But I want to get a few. I've heard some correct answers, so take the answers or those hypotheses of how to do this. And I'll take those, and then I'll work through how you would design an experiment. Always, when designing an experiment, you need to think to yourself, I would have this where I expect it to work. I would have this where I would expect it not to work. And so you're adding and subtracting things to kind of get to where you can look at all the data together and definitively say protein X is a helicase or not. Okay, so what did I hear first? Yes.
So our idea was kind of if you started out with your proteins, or you started out with your DNA and you ran on the gel to see the different sizes and know what you have. And then in a test tube, you add the single-stranded DNA and you add ATP in your protein. If you take the results of that and you run it through a spectrophotometer, you should see different levels of the protein, or different levels of the single-stranded, double-stranded DNA. If it was positive, then helicase, you would have more short single-stranded.
Okay, so basically you would either with or without your protein look at how much single-stranded versus double-stranded DNA you have, right? Pretty much. Okay, so that's one. Yeah. Same. So that was looking at based on OD, right? Or not OD, but yeah. What about using the gel? What would you expect? Yeah.
We use the gel as a final product to test whether or not the protein split the double-stranded DNA, and based on the weight of the DNA on the gel, you can tell whether or not it's a helicase or not. That's just one.
Right.
So there should be more dense in the bottom because the smaller ones will migrate further away.
So you would see a difference. So let's go through exactly what we would expect to see. Does anyone have anything else to add? So looking at the differences, without the helicase, you should have double-stranded of a certain weight, which is small plus large. With the helicase, you should have small and large separate, right? Okay, yep.
Just a question. Is it possible to run one with a single strand and run another one with a double strand?
So is it possible to run one with single-stranded DNA and one with double-stranded DNA? It is important to have a control if you're running a gel where you would have single-strand only and double-strand only, so you can at least know what it looks like, but that would be a control.
No, I mean like one single strand with the protein and then the double strand with the protein. Because the single strand
Well, nothing
would
should
not
have.
exactly.
Yeah, so you're saying as a control, run single-strand alone with the protein, and then run the protein with double-strand. So that's another way to do it. Yep.
Or as a control, you would run the spectrophotometer and do a gel.
Without the
Yeah,
helicase.
do both, and then you get your result.
Right, so normally that's what you do. Right, so yeah, you can do both spectrophotometer and look at it on the gel. And that's usually what you do when you're studying something is you look at something 10 different ways and make sure all the data is in line with your hypothesis. Yes?
Can you have control without the unknown protein?
Yep. A control without the unknown protein. So I'm going to go through it with you exactly how you would approach this for those of you, because for some of you that you're like, oh yeah, and then for some of you you're like, I don't even know what to do here. And so if this comes up on an exam, this is how you would do it. Well, first you would come up to me and be like, I don't even know how to deal with this. And then I would talk you down, and then I would help you by at least putting you in some sort of direction of how to think. But, so you know you have, okay, so the things that you have is single strand DNA short, single strand DNA long, and you know if you incubate them together you would end up with double strand DNA of short plus long size. Short, why not? Well, either way, short plus long. Okay, so you've got that. And you have ATP, because you know, because you did your reading and you're smart, ATP is required. Okay, so the first thing you would do is design your experiment, making sure you, oh, and protein X, duh. Okay, and so always remind yourself, what question am I asking? Is protein X a helicase? What do I want to see to prove it's a helicase? I want to see double-stranded DNA become single-stranded DNA. Okay? So normally you would start by having, so here would be your different tubes, right? One would be, single-strand DNA plus double-strand DNA alone. 2 would be single-strand DNA plus double-strand DNA, bless you, plus protein X. 3 would be single-strand DNA plus double-strand DNA plus X plus ATP. And single-strand DNA, single-strand, sorry guys, these are both single-strand DNA. One is just short and one is long. So this would be S for short, L for long.
But some of them
Because in one, you should just get, right? That's what it would look like in your tube, right? Oops. 2, oh, I see what I did. Oh no, I'm good. 2, what would you expect? Same thing, right? Because there's no ATP. So it's a control showing it's ATP dependent, right? And so 3, what would you get? Right. Because you would expect your helicase would come through and unzip it, right? And so if you're running a gel, and I guess we can come up with more controls to put in, but we'll keep it simple for this. If you're running a gel, so first of all, if you do spectrophotometer, this should look the same, but this one should look different, right? Because single-strand DNA absorbs differently from double-strand DNA. So that's plus minus, right? It's either there as single-strand or it's there as double-strand. If you're running a gel and you load at the top, each one, here you should get I don't know what size it is, but let's say it's here. Here you should get a band here, but here you should get that. Oops. Okay, and so ideally together that data all makes sense. Now the next thing you do is, I guess, chop out the helicase domain and show that it doesn't do it anymore. Okay, so that's how you would go about designing an experiment. Now mind you, it takes a lot of years to do this, and I wouldn't give you something this difficult on the test, but I think by getting your brain to start thinking this way, the simpler things on the exam will be very easy, hopefully. Any questions about how we went through this? Yes?
Can you give me your concentration of ADP and free phosphate to us? Because if you know that's ADP relative, like
Okay. So I just got the comment. Can you monitor the concentrations of ATP and ADP or free phosphate or whatever? Will that tell you something? That would be data that would support your hypothesis, but would not directly answer. Because all that says is, does protein X hydrolyze ATP? Now, anything with an ATPase domain will hydrolyze ATP. And so you would probably do all these things and say, and look, ATP is being used up. And look, if I chop out this, you know, that's how you get to a research paper. It's going at like 10 different angles. Okay. Any questions about this? Did everyone like that? Okay. I'm getting to do a lot of writing today. Okay, so that was our practice problem. Oh dear. Okay. Okay, single strand binding proteins. Called SSB in bacteria, called RPA in eukaryotic cells. They stabilize separated DNA strands. Right? Because if you don't have single strand binding proteins, what's going to happen? Regions that are complementary are going to base pair in the single strand DNA, and then you have a problem when DNA polymerase wants to come through, because you suddenly have regions of double strand DNA. So these single strand binding proteins are going to coat the DNA and keep it single stranded, and then as the polymerase comes by, they'll release from the DNA. Okay, so let's talk about topoisomerase. Oh man, I have a demonstration for topoisomerase. So I'll talk about it briefly, and then we'll start next week with the demonstration. My favorite, when I was taking MolBio in grad school, the professor came in with a telephone cord. Like, remember, I don't know, maybe you guys are all too young. Who in here is old? Do you guys remember when, like, in your parents' house, like that cord would get all wound up in itself? Y'all supercoil? That's right. So that's what supercoiling is, right? And with the telephone wire, you're not dealing with double-stranded things. But you can imagine if you have a helix and you separate and pull it apart, it's going to supercoil. And that's where my demonstration comes in. Anyone who knits, go home, grab some yarn, and pull it apart if there's two pieces, or separate it into two chunks, and you'll see it'll supercoil. But when I was taking this, the professor brought in a telephone cord and like had another student come. They did this demo, and I was like, oh. So that's going to happen, and you don't want it to happen. So that's why we have topoisomerase. And there's multiple types of topoisomerase. And basically what they do is they remove supercoils, and those supercoils are created by the unwinding. Supercoiling causes a lot of torsional stress. It's not good for the DNA. It's not good for replication happening. You can imagine if you're pulling something apart and it's coiling up on itself, you're going to get to a point where you're not going to be able to replicate anymore. So you need topoisomerase. So there's a few different types of topoisomerases. There's one kind that is topoisomerase 1B, 1 beta, I believe. But basically, type 1 topoisomerase, it rotates the DNA. So what it does is the enzyme comes in and it has this tyrosine in its active site, and it does one of these things where it's got the OH group and it attacks the phosphate backbone and creates a bond there. And by doing that, it makes a nick in that one strand of DNA. So it's not a double strand break, it's only one strand. And it's got all that torsional stress already there. And so it just spins around. So that one's kind of an easy one to visualize because it makes this nick and then this is going to spin back. And then it's able to kind of attack back on the enzyme and release itself, and so create the bond again. No ATP is required. It's the torsional stress that's already built up in the DNA that powers that rotation. Oh dear, yes.
So when it's moving, like making all the, is it possible to throw away, like push away those histones? Like, because it's. the
Don't
speed-
forget, this is happening during DNA replication. This is happening, so the question was, is it possible to move away all the histones and all that? And so during DNA replication, your histones are being removed from the DNA in areas of active replication. So they're not too much of a problem. And the topoisomerase is usually just a little bit ahead of helicase. Make sense? Okay. Yes.
I don't know if it's just me, but is it removing the OH? And if so, if it's removing the OH, how do you recognize 3' OH?
It's not removing an OH. What it's doing is, so this is a tyrosine which has an OH in its side chain. And so that OH is going to attack the phosphate and then make a bond there. And so you've got an OH here and you've got that bond there, and so there's your nick. But then it's going to go back and attack and create the bond again. So you're not actually losing anything. Yeah. Any questions from this side of the room? Nope. Did I see hands up over here? Okay. Okay, so then there's type 2 topoisomerase, and it relieves torsional stress by introducing a double strand break in one segment and allows an intertwined double strand segment to pass through. So this is double strand break. So this is where you're not dealing with one double helix, you're dealing with two double helices that are all tangled up, and it'll create a break in one and pass through the other. So if you keep an eye on the blue and the pink, that's two different double helices. And what's going to happen is, right now the blue is on top of the pink, and after the enzyme works, the blue is on the bottom of the pink. And so what it's doing is it's basically creating a nick and passing the blue through to the bottom and untangling it that way. So for this one, ATP hydrolysis is required. And then there are also type 1A topoisomerase enzymes that use strand passage. This is called strand passage. Makes sense? It's passing a strand through a break. Type 1A enzymes also use strand passage, but it's the passage of one strand of DNA through a break in a single strand, but we're not going to worry too much about that right now. So then again, this is just another way of looking at this type 2 topoisomerase. This is just out of a review. Here's your topoisomerase, and it's going to bind to one strand of the DNA, and then it's going to bind to the other strand of the DNA, and then it's going to nick it and pass it through. Same thing. Okay. How are we doing on topoisomerase? Good? Okay, I think I'm going to stop here because my throat hurts. Yes?
You usually have